The origin of life. The origin of life has never been duplicated, although we have come close, and it was attempted many times. Some of the first scientists in the 1920s tried to figure out how life began. One of those, well, two of those, were Alexander Oparin and J.B.S. Harding. They hypothesized that life on Earth began in an acidic primordial sea. This began known as the Oparin-Harding hypothesis. In this hypothesis, a series of chemical reactions caused life to complicate into coastrates within the aquatic environment, then created life. Now, in early 1953, Stanley Miller and Harold Urey were inspired by the Oparin-Harding hypothesis and brought it to life in the Urey-Miller model. This is where they recreated the Earth's early environment, which was an acidic primordial sea and Earth's highly dense atmosphere full of volcanic like ash and lots of chemicals, heavy chemicals.
In the 1920s, Oparin and Haldane hypothesized that the atmosphere of the primitive Earth contained gases such as nitrogen, ammonia, methane, carbon dioxide, water vapor, and hydrogen. Oxygen did not become a part of the atmosphere until the advent of photosynthesis millions of years later. It was further hypothesized that under these conditions, organic molecules could be formed if there were a source of energy such as electricity from storms or ultraviolet radiation from the sun. In 1953, Stanley Miller and Harold Urey tested this hypothesis. They constructed a closed system with a reservoir for water, sampling ports, a chamber for the input of electrical energy, and a condenser to convert the water vapor back to a liquid. The researchers then added water, hydrogen gas, methane, carbon dioxide, ammonia, and nitrogen. The water was heated to form a vapor, and electrical sparks were generated to mimic lightning. This apparatus simulated the possible conditions of the early Earth. When the water was analyzed after 1 week, they found simple carbon compounds such as formaldehyde and hydrogen cyanide. These compounds then combined to produce formic acid, urea, and amino acids. In similar experiments carried out over the next several years, various organic compounds have been synthesized. These experiments showed that simple organic compounds, including the building blocks of proteins and other macromolecules, could be formed from gases present in the primitive atmosphere with the input of electrical energy.
Even though they could not recreate life, they did find organic molecules within their solution. This was a great step towards recreating life in a lab. So in the early 1960s, Sidney Fox took this experiment yet another step closer. He took the Miller-Urey model and placed the solution that he had created on different surfaces that he might happen during early Earth's life. So he recreated things like as rain would come down, it would land on different layers, like really hot sand or dry clay or lots of types of rocks that you could have seen in early Earth's life. And after this experiment, Sidney discovered that the aggregates spontaneously formed into proteins. This is very significant because proteins are the building block of life. This is the closest that we've ever come to recreating life in a lab, and this is very important towards our origin of life. Alright, hi, this is Alfredo, and that's Stephanie behind the camera, and what we're doing today is we're going over the origin of life. So for this lab, we're going to need a few materials, and one of the materials is the gum arabic solution. Now, this is a macromolecule. It's a carb we're going to be using for our lab. And next, we will need a gelatin solution. This is our protein, and that's cold. It's gotta be really cold. Next, we'll need a pipette, a microscope slide, and a microscope. So in this lab, we are going to try to recreate coastermates.
So what we're going to do, you have your gelatin solution here and your gum arabic here. You're going to take some gum arabic solution and put it into the gelatin solution. So put it in. And then you're going to mix the solution. This is a coacerbate solution. Gently like this, not vigorously. And then you're going to observe what you see. You're going to ask what color it is. Is it clear or cloudy? Is there precipitate in the solution? And after you do that, you can make some observations. And based on what your factor is or whatever you're testing within your own lab that you designed, you're first going to put the coacerbate solution on the slide using the pipette. And then after you do that, you're going to use a cover slip. So there's no air bubbles or anything, so you can observe it correctly. And observe it first to see how many coacerbates and what they look like underneath the microscope.